Hello, my name is Deanna Burkhammer, and I'm going to show you what we did in Bio 1A at Holy Names University with Dr. Apollon. Hunting for microbes, macromolecules, and is there bacteria in my lunch? This experiment is the beginning of our research for the term. On January 25, 2016, we picked two objects from our lunches that we swabbed for bacteria. For my experiment, I swabbed a piece of lettuce and a lemon for my lunch. In this experiment, we will be looking for evidence of life in our food, and we will be culturing microbes. Microbes are generally found everywhere and usually harmless. They actually play a pretty important role in chemical and energy cycles. We want to determine if there are any microbes living in my lunch. If microbes are present in my lunch, then there would be several colonies in my agar plate. If microbes are not present in my lunch, then the agar plates will be empty. Materials: 1 tube of sterile water, 2 sterile cotton swabs, 2 sterile nutrient agar plates, and 2 samples, lettuce and a lemon. I swapped sample number 1, lettuce, and sample number 2, lemon, and I rubbed around a bit, pressed the swab into food. Then I lightly painted the entire surface of one of the NA plates with sample 1, then sample number 2 swabbed by wiping the cotton swab across the surface. Once done with that, I placed all plates inverted in the rack to be incubated at 37 degrees Celsius for 24 hours. After 24 hours in the incubator, I examined how my colonies grew. I analyzed the sample number 2, lemon, and had less colonies than sample number 1, lettuce. Lettuce had clearly 50 plus colonies on the agar plate, while lemon had about 2 large colonies. The agar plate on the left represents the lemon. The lemon had a circular, irregular in shape, curled margin, flat elevation, and it was large in size. Only one dot was on the disc. Texture seemed smooth, and the appearance was dull. Pigmentation was white cream and translucent. On the lettuce, on the right, the lettuce to me appeared to be circular in shape, entire margin slightly convex, elevation with multiple moderate and small colonies throughout the disc. The texture was smooth, and the appearance was glistening. Pigmentation was cream, and optical property was opaque. This leads to the next lab, hunting for microbes 2, colony morphology and isolation, diffusion, and antibiotic resistance. This is the lab's purpose, is to isolate and grow a pure microbial culture. If I am able to isolate a pure culture, the microbes will have colonies on the agar streak plate. If I am unable to isolate a pure culture, then there would be no colonies on my agar plate. Materials: Bunsen burner, metal inoculation loop, 2 new sterile NA plates. Within this lab, I divided the agar plate into 4 sections and created a streak plate for the lemon and lettuce bacteria. I used a new sterile NA plate and lightly dragged my loop several times through the first quadrant to spread the bacteria all around that region. After swabbing all 4 sections of both agar plates, one with the lettuce sample and the other from the lemon sample, we incubated the plates for 24 hours. After 24 hours, we took our streak plates out of the incubator and I observed the colonies for my streak plates. Noticing that the growth only appeared on the first out of 4 sections, I swiped. On the lettuce, the shape was irregular, the margin was lobate, elevation convex, size large, texture rough, appearance dull, pigmentation tan, and optical properties opaque. On the lemon sample, the shape was irregular, the margin was undulate, the elevation was raised, the size was large, texture smooth, appearance glistening, pigmentation cream, and optical properties was translucence. With the streak plate, we noted that the morphologies differ from the first time these microbes are growing. Now the colonies appear to be larger with the second experiment. Now on to the next lab, diffusion and antibiotic resistance. But did you know that antibiotic resistance is often found on a piece of DNA called a plasmid that can be passed from one bacterium to another? Each plasmid usually contains information so that the bacteria make a protein that allows them to survive even in the presence of an antibiotic. In this lab, we're covering antibiotic sensitivity. It's how effective the antibiotic is at preventing bacterial growth. The more sensitive a bacterium is to an antibiotic, the less of that antibiotic is needed to prevent bacterial growth. The purpose of our next lab is to test the sensitivity of known bacteria cultures to various antibiotics. Let's review our hypotheses for this lab. If these bacteria are sensitive to antibiotics, then there would be a zone of inhibition around the antibiotics. If these bacteria are not sensitive to antibiotics, then there would be no zone of inhibition. But if these bacteria show different sensitivities to antibiotics, then there would be some antibiotics that will have different size of zone of inhibitions, or there would be some that have none at all. For this lab, we'll have a Bunsen burner, metal inoculation loop, 2 new sterile plates, and 2 liquid bacteria cultures, the lettuce and the lemon, and a variety of antibiotic discs. I swapped sample 1 lettuce and sample 2 lemon, and I rubbed around a bit, pressed the swab into the food. Then I lightly painted the entire surface of one of the NA plates with a sample 1 sample lettuce bacteria, then sample number 2 sample of lemon bacteria, and swabbed by wiping the loop across the surface. Then we carefully dropped the antibiotic disc. We dropped one of each antibiotic type onto the bacteria and spread the different antibiotic discs out so they are not overlapping. Then we placed both of the plates inverted in a rack and incubated at 37 degrees Celsius for 24 hours. After we incubated our plate for 24 hours, we were able to conclude that vancomycin, chloramphenicol, ciprofloxacin, and tetracycline were all resistant to the bacteria in lettuce. However, tetracycline was inconclusive. Vancomycin had a diameter of 2 millimeters, ciprofloxacin had a diameter of 0.7 millimeters, chloramphenicol had a diameter of 3 millimeters, and penicillin had a diameter of 0.8 millimeters. For the lemon sample, I was able to conclude that there were no antibiotic resistance in VA, CIP, and C. Although penicillin having a 3 millimeter diameter, I was able to say that there was antibiotic resistance. There were some errors since the antibiotics were moved around, which made tetracycline inconclusive. I was able to support my hypothesis that if microbial resistance were present, then there would be a zone around the planted antibiotic would occur. Some antibiotics were moved around, and that could have affected the results. To improve or fix my experiment, I would make the antibiotics more secure in an area further away from one another, as well as trying new antibiotics or test lettuce and lemon from different places around the world. In the next part of the lab, we are going to use microscopes to view our bacteria samples. The following materials are needed: 2 sterilized slides, 2 sterilized cover slips for the slides, alcohol iodine microscope, Bunsen burner, and a metal inoculation loop. We used our previous samples of bacteria in order to make the slides of lettuce and lemon. Bacterial cells are usually colorless because of the cytoplasm. For the most part, it's transparent. Since the bacteria are colorless, it is almost essential to add a stain to make the bacteria more visible. Once stained, cell morphology can be observed. Using aseptic technique, we smeared the samples. Then we let the preparation on the slide dry on the slide dryers atop the BacDye incinerator. Once done, we held the slides smear side away from the heat near the mouth of the BacDye incinerator for a few seconds, and placed the slide on the microscope and observed both slides using all powers of our microscope, including oil immersion. These are a few pictures from the lab. First, we sterilized our loop, took the sample from the bacteria, placed it on the slide, stained the slide, and then viewed our bacteria through the microscope. These are actual pictures of the lettuce and lemon bacteria. Unfortunately, after trying to observe both bacteria from lettuce and lemon under the microscope, we were unable to see any bacteria on my slides. The lettuce slide was too dark, and my lemon slide was contaminated with cheek cells due to me blowing on the slide trying to dry it faster. In our next lab, we're going to do genomic DNA isolation. The purpose of this lab is to extract the DNA from the lettuce and lemon samples in order to detect bacteria and viruses in the environment. These are the few materials we're going to be using in our next lab: centrifuge, bacteria samples of lettuce and lemon, enzymes, and rubbing alcohol and buffer truss. I used my lemon and lettuce sample, then lysed the cells, used an enzyme to break down protein, precipitated the DNA with ethanol, washed the DNA, and then resuspended the DNA. Yay! We made a DNA extraction. Now the presence of DNA can be confirmed by the electrophoresing on an agarose gel containing ethidium bromide or another fluorescent dye that reacts with DNA and checking under a UV light. In our experience for PCR, gel electrophoresis, and bioinformatics, we used our bacteria in these experiments to isolate the gDNA, measure, and blast to identify the bacteria isolated with a sample from our lunches. In the PCR lab, we're going to need the following materials: pipette, pipette tips, PCR machine, bacteria samples, in my case it's lettuce and lemon, 25 millimeters of magnesium chloride, Taq polymerase, primers, and extracted DNA. So let's get to those PCR methods. We used 2 tubes that contain 30 microliters of master mix for the 16S rRNA gene amplification. Next, we added 1 microliter of our gDNA sample to every primer solution. Then we used 2 PCR tubes and added 25 microliters of gDNA solution and placed the tubes in the thermal cycler, which went through denaturation, annealing, and primary extension. This isolated our gDNA. After we used the PCR, we analyzed our PCR products by agarose gel electrophoresis, similar to what we did earlier with our food dye. For gel electrophoresis experiment, we're going to need the following materials: a weigh station to weigh out agarose, flask, green dye, TAE buffer, gel electrophoresis apparatus with a 10-tooth comb and power supply, tubes, DNA marker, gel loading dye, Brofenol blue, Pitman and tips, purification kit, primer and DNase free water, and sterile DNase free water. With our gels that we already made, we used them to prepare the gel chambers and took the tube and labeled them. Then we took out the PCR product and combined a gel loading buffer in the labeled tube. Next, we loaded all 7 microliters into a well of the agarose gel, repeated and recorded the contents of each. Then we loaded 3 microliters of DNA size marker into the last of the first well. Once our PCR samples were in the gels, we ran the gel in order to separate DNA molecules on the basis of size and shape. These DNA molecules became visible as fluorescent bands in the gel when placed on the blue light bed. The brighter the band, the better the DNA yield from our PCR. We measured the distance from the well to each of the known bands in the ladder and the distance from the well to our PCR band product. Now, we're going to use the PCR product, purify it, and prepare it for the sequencing. We took 1 tube of DNA binding buffer and added 15 microliters of my PCR to this tube, then transferred all the tube contents to PCR purification tube. We then spun this column at 13,000 RPMs for 30 seconds, dumped out the contents, added a buffer to the contents, and then added a wash buffer to the column and spinned it again for 30 seconds. Dumped the contents of collection tube and replaced it with a clean tube. Then added sterile DNase free water to the column, incubated for a minute, and then spun again at 13,000 RPMs for 30 seconds. Now, my purified DNA is now ready for sequencing. In our last lab, we had samples that the instructor prepared for us to blast through Finch TV and the BLAST website. At the end, there were only 8 microbes chosen from the class lunch samples, which were identified by DNA sequencing analysis. Then we purified the PCR DNA product and prepared our DNA sample to be sent to a local DNA sequencing facility for analysis. Afterwards, we received the DNA sequence, we blasted it to find the strand of bacteria isolated. This is the data that we got from the isolated bacteria. From the results I received, I was able to support my hypothesis that if bacteria was present, we would be able to isolate the bacteria from our lunches. Although some bacteria were isolated and identified, there could have been mistakes through handling and going through the steps in order to blast, which could have ultimately affected the results. To improve my experiment, I would test similar foods from different environments and go through the same process or similar process to see how the results would happen.